is Eric Henry, and welcome to video number 1 of 10 of the cell cycle series. This video is titled Control Mechanisms in Cancer, and in it we will discuss the cell cycle itself and how highly regulated it is. We will also see what happens when the regulations are not met. Now, this is a picture of the cell cycle, and the cell cycle is basically a process in which cells divide. Here we are looking at mitosis, in which somatic or body cells, like skin cells or bone cells, actually divide. And the cell cycle actually has 6 parts, and it goes through interphase, which has the G1, S, and G2 phase, prophase, metaphase, anaphase, and telophase or cytokinesis. Now, in the interphase, the G1 phase is where the cell is growing, and then the S phase is where the cell, the DNA inside the cell is replicating and turning chromosomes into sister chromatin, which is very important for later. And then in the G2 phase, the cell continues to grow and gets ready for prophase. Now in prophase, the sister chromatin are being more tightly spun so that under a light microscope they're very, very easily seen. And this helps with after the replication, it'll help prevent any problems, any crossing over that might occur. And then in metaphase, the sister chromatin are lined up along poles, and spindles start to occur. And these spindles will come and start attaching to the actual legs of the sister chromatin. And this will set up for anaphase. And in anaphase, the spindles pull apart the sister chromatin at the centromere and start to pull apart into either pole, I guess you could say. And then in telophase, or cytokinesis, the cytoplasm and all the rest of the organelles will start dividing, creating 2 identical daughter cells. I guess, as you can see, this process is very complicated, but checkpoints are ensured along the way at the G1, G2, and M or mitotic phase, so that the cell is very fluid and no mistakes are generally made. Now the cell cycle, as we talked about, is highly regulated, and a way they can be regulated is through cyclins, which are proteins in every single cell of your body, and CDKs or cyclin-dependent kinases, which are also found all through the body. But CDKs are inactive proteins that are actually activated by cyclins. And then another way that the cell cycle is regulated are through checkpoints, which we just talked about. The G1, G2, and M checkpoints are all set up so that the cell cycle has a place to regulate and make everything fluid. And then along with checkpoints, MPF and PDGF, which are 2 proteins, are also used to help kind of signal the cell, like stop and go signals, to make sure that the cell knows yes, it can go on, or no, it does not need to replicate now. And then we also talk about cancer, which actually happens due to a lack of regulations in the cell. Okay, so we're first going to talk about internal controls, which are also labeled as checkpoints or sometimes stop and go signals in the cell. So here we see a picture of a time lapse of interphase, actually, and on each point of the process in the G1 phase, you can see this G1 checkpoint. Right after the G2 phase, you can see the G2 checkpoint and the M checkpoint right here. So basically, in order for a cell to get past the G1 phase into the S and G2 phase, it must pass this initial G1 checkpoint. The G1 checkpoint is also a very, very crucial checkpoint. It is known for, it's very known as a restriction checkpoint, because if the cell is given a stop sign saying like it cannot go past this checkpoint, the cell will actually go into a phase called G0, which is where the cell is not dividing and it can't divide, it can't grow anymore. This is typically where you find nerve cells, because nerve cells, once they are broken or ruined, basically, they can't create new nerve cells because they are in this G0 stage. They will never divide. So, as you can see, the 3 checkpoints right here. The G1 and G2 checkpoints are regulated by proteins or kinases in order to give that stop and go signal. If the kinase is there, then it'll give a go signal, and if the kinase is not, it'll give a stop signal. The G2 phase is actually regulated by one of the proteins we talked about earlier, the NPF protein, and we'll talk about that a little later and exactly what that does and everything. And then the M checkpoint is just the final checkpoint. Interphase goes through in order to get past interphase and go into the prophase and metaphase and just continue that phase. Along with the internal controls, we also have external controls, and internal and external controls provide these stop and go signals as we've talked about for these checkpoints at the G1, G2, and M phase. And so they can either say, don't go on, don't divide, or yes, everything's ready, go on and divide. We will first talk about NPF, and NPF is actually the maturation promoting factor. And it's basically, as we've said, it's the factor that must be there in order for a cell to pass the G2 phase. And if it's not on the outside of that cell and if it's not detected, then the cell will stop its cell cycle immediately and does not continue. And then PDGF, which is platelet derived growth factor. PDGF is actually the protein involved in the G1 checkpoint. And you can commonly see it when you get a cut or you get basically an open wound. And the PDGF, if it is seen on that G1 factor, or the G1 checkpoint, will actually allow these cells to start dividing and basically getting a lot bigger. And you see that with the skin tissue around the open wound, because if that PDGF is there, the cells will start dividing and start closing up that wound a lot more quickly than it should. And this helps regulate, these two proteins help regulate the cell cycle, because if neither of them were found, the cell cycle would go crazy and just keep dividing and keep replicating. And instead of, if they weren't there, it would tell it to stop, so it would not do that. Typically, lack of these proteins and other different types of proteins cause the cancer that we're going to talk about sooner or later, because the cells just keep dividing and keep replicating. Okay, cyclins and CDKs, or cyclin-dependent kinases, are found in every single type of cell, and they are also, like NPF and PDGF, are proteins that regulate the cell cycle. In order for a kinase or a CDK to be active and to be able to be used in a stop and go signal, it must be attached to a cyclin right here. And if it's not, it's basically useless for the cell because it isn't an active protein. Okay, now cancer. These are actually cancer cells, and cancer is the lack of regulation in a body or in an organism or in the cell cycle. So when there is no regulations, the cell just keeps dividing and dividing and dividing, causing tumors and other really serious problems, because if cells can't stop dividing, these tumors form, causing other health issues, like really, really severe forms of cancer. Now, malignant versus benign tumors are very, very common. Malignant is actually the cell cycle will keep going, and the cells will just keep dividing, and it's growing, and there's no way to stop it. And then benign, it means it has grown, but it has already stopped growing, so it's no common threat. And yeah, that's it for the cell cycle and regulations. Thanks for watching.